This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs. Also, make sure to check out and subscribe to our YouTube original channel, UCTV Prime, available only on YouTube.
I'll talk a little bit about one aspect of ADHD that has seemed to us to be an entryway into the confusion that is this common disorder. And we'll focus mostly on the use of what's commonly called resting state, but that's probably a misnomer, but a mode of measuring brain function without a task that seems to be giving us some exciting clues. And one of those has to do with what's called the default network interference hypothesis. And one of the ways in which we're exploring this is to aggregate samples from investigators literally around the world. And I'd like to propose that that's something that we should look forward to doing more and more. So about 10 years ago, Rosemary Tannock and I were musing about what did we understand about ADHD? And we're thinking about the decades of neuropsychological work that have been done trying to get to a test. Everyone wants some kind of objective marker, some way of measuring that would be either like a blood test or a blood pressure test or something that doesn't rely simply on opinions. And it's been 30, 40 years of disappointing work. And in part, that has to do with the dramatically substantial inconsistency of performance of individuals with ADHD who will do beautifully on a test one day and terribly the next, or even that same day for part of the time. They'll go in and out. And so we thought about how that inconsistency perhaps was an important clue. And we suggested that not based only on our own observations, obviously, but on previous literature that had started to highlight that. And one of the things about that concept is that it's relatively easily measured if we track how long it takes someone to push a button, response time. And there's a finite amount of time. You can't respond in 0 time because it takes about a quarter of a second for the information on the computer screen to get through your eyes to the visual part of your brain, and then for that information to be processed and eventually make it through to the motor centers, etc. So there's always a minimum amount of time required, but there isn't always a maximum amount of time required. It could take longer. If you're distracted by a cell phone ringing, then you come back to what you were doing, then it'll take longer on that trial than in other trials. And so when we measure a series of response times and sort of put them up next to each other, we typically see a kind of ski hill configuration. So it's sort of steep on this end and then long and sloping on that end. And what a number of investigators had been observing is that the ski hill was longer for individuals with ADHD. So they would tend to have more of these very long, prolonged response times. And this was true from data aggregated from the MTA study, which was a large multi-site study. And we also found this in data from a postdoc at the time, Anouk Shears, who had used a standard Erickson flanker task in kids with ADHD, where they're told just to pay attention to this arrow in the middle and ignore the others. But sometimes the arrows are all pointing in the same direction, other times they're conflicting, and other times there's just a rectangle that doesn't give you any information. And what's nice about this test is that you can predict very well that individuals, it's easy to do, first of all, you just have to say left or right over here, but it takes between 50 to 100 milliseconds more on average to respond to these stimuli than it does to that one. So you can really tell if people are sort of tracking the task or not. And Anouk was getting the kids to do this every 3 seconds. And so this seemed like a good place to examine consistency and see whether or not that was systematically varying. And here's a couple of illustrative cases. This is what a control looks like, and we've done a little bit of smoothing here, but more or less there's going to be some up and down jitter because, again, it takes longer to respond to those conflicting trials than to the non-conflicting ones. But here's a typical child with ADHD. On average, they're about the same, about half a second. But here, this one took almost 3 times longer to do that same task. And here it's twice as long, and there it's almost twice as long. So those we take as indicators of lapses of attention. That the child knows every 3 seconds I'm going to have to respond, but occasionally they're having to bring themselves back sort of from a longer distance. Now, these data can be analyzed in a number of different ways. And one of those is by assuming that, in a sense, it's like a piece of music. And so we can do a frequency analysis, which is the same thing that happens when people package music into an MP3 file or a CD, which is you break up the musical spectrum into frequency bands. And these are the frequency bands along the bottom axis. And then you can determine how intense or how loud the frequencies are at any given frequency. And in this case, there's really no particular frequency band that stands out in this child's record. Whereas in the ADHD child, there's a very clear peak at about a 20th of a hertz. In other words, it takes about 20 seconds for one of these cycles to go through its full up and down oscillation. But when that child was given methylphenidate, you see the darker tracings, and you see the tracing of the frequency analysis. In other words, that variability was abolished and brought to the control levels by Ritalin brand or methylphenidate. And so that struck us as interesting. And around that same time, there was a group in Germany, Christoph Klein and colleagues published several measures, go, no go, stop task, continuous performance task, and NBAC. And regardless of the task, the variability of the children's response times, measured in a couple of different ways, was always the best way of discriminating the children with ADHD from the controls. And in fact, when statistical analyses were done, called principal component analyses, about 2/3 of the variation in response times could be assigned to this single focus of variability. Jona Kunze and her colleagues at the Institute of Psychiatry have done a series of studies going back to 2001 showing that, in fact, this is the only neuropsychological trait that seems to be heritable and very robustly heritable. So again, it speaks to genes being involved in how it manifests. And what's interesting is that this kind of variation in response times frequently masks as the inability to do executive function tasks, because what you get is less average performance, and it looks like they don't get it, but they get it very well, and then they don't get it periodically. And so we've been chasing that idea down, and in biological psychiatry, we published a paper in 2008 in which we took an independent sample and gave them the same task, and again measured those frequency differences and found that there were more variation, especially in that very low frequency range in the children with ADHD, and that the more of that energy they had, the more likely they were to make various kinds of errors, here omission errors. Catherine Johnson at Trinity College in Dublin had found a similar observation in about half the kids with ADHD. The ones that were impaired in terms of making more errors had clearly greater intensity of that low frequency variation, and with different kinds of task. It's called a sustained attention response task. And so we recently did both that SART and the Erickson flanker. This is in a group of Italian children, and again found that using both methods of measuring frequencies. I won't go into the technical issues, but one's Morley wavelet-based, and the other is the Fourier frequency transform. In both cases, for both tasks, we see greater variability in the individuals with ADHD than controls. Many of these kids unmedicated ever. And again, when we separate them out by the impaired versus the unimpaired, we see that some kids with ADHD look just like the controls, and others are very substantially impaired, these green sort of shadings there. We're not the only ones. Again, the Johnson's group and others have found this. Not everyone gets it, so there are issues perhaps about tasks, etc. But it's compelling enough that we've been able to proceed with this. And around the same time, in the world of neuroscience, Yuri Busaki and his colleagues who were really interested in oscillations and fluctuations in neurons at very high frequency rates, observed a number of things. One is that it doesn't matter what size brain an organism has. It could be a mouse or a human or an elephant. The same frequency bands are present if you measure their EEG. And so it's not about the size of the brain, it's really about the molecules and the cells and the circuits that are present, and those are really maintained across the whole evolutionary spectrum. So it's a reflection of intrinsic cellular and circuit properties. And he stated that the synchronous activity of oscillating networks is the critical middle ground between the neuronal activity, which happens in thousands of seconds, and behavior, which happens over seconds and minutes and hours. And another interesting observation was published in a little-known journal called Thalamus and Related Systems, which is that when they took the standard frequency bands and plotted the midpoint of alpha, beta, theta, gamma, etc., and placed them on a logarithm scale, each one of these was one natural log unit apart. And I've always loved the mathematical sort of symmetry of that. And so that gave them a kind of rule that they could use to define even lower frequencies. And so they don't have names for those, so they became called slow 1, slow 2, slow 3, slow 4. And slow 4 is where we've been focusing, where we saw those fluctuations of reaction time. And slow 4 is also of interest because it turns out to be the rate at which our brains are fluctuating. So this is an art project that was created by one of my, at the time, students, Daniel Margulies and his colleague Chris Sharp. But it's real fMRI data of one person listening to his favorite piece of music, which I'm not playing for you here, but if you go to this web link, you get the whole video, which is the listener listening to The Rite of Spring by Stravinsky as the music is playing. And so you get into all of these issues about, the art experience, and it almost looks as if the brain is dancing at times when you see this music. But this is also a reasonable portrayal of the real-time fluctuations of the brain in obviously color-coded by the computer, so that the red warm colors are indications that the bold signal, which is related to blood flow, is greater than average, and the cool colors are less than average, and green is average. And what used to be thought is that these fluctuations are basically random noise. But I think just observing this for 90 seconds, which is what we're doing, shows how systematic and how lawful these correlations are. And I can personally watch this for 35 minutes without sort of feeling bored. But I won't make you do that. It's just 90 seconds. But this is very striking, very robust, and we don't understand why. But one of the pioneers of this phenomenon is Mark Rakel, who has been focusing on it for, since 2001, and recently wrote an article on the brain's dark energy describing this, because Rakel and others assert that the majority, could be as high as 80% of the brain's energy, is basically expended in the maintenance of these relationships, completely independent of the thoughts that the person's having or the actions that they're manifesting, that it's this background activity that's always ongoing, unless we're brain dead, that consume a great deal of energy and therefore must have quite a bit of biological importance. But like an iceberg, which is mostly below the surface, we haven't been paying attention to that because it's been very difficult to sort out how to measure that. So that's sort of his metaphor, among others, for this phenomenon. And the field has kind of taken to this, although, as always, it took a while. So the first paper that really examined this phenomenon was in 1995 by Bharat Biswal, who was a graduate student at the time. And Bharat, to this day, is a very modest and meek individual, and has yet to be funded for doing intrinsic functional connectivity studies, but has managed to be an imager with funding in other areas. But his paper was published there, and there was very little response to it. A couple of studies that sort of built and followed on that, because he was working with Jim Hyde, who was, again, one of the pioneers of imaging. But it was in 2001 that Rakel published a couple of papers, including one in the Proceedings of the National Academy of Sciences, because he's a member of the Academy. And so one of the privileges is that you get to publish your own work a couple times a year, just because you think it's important. I didn't have the guts to ask him directly, but I suspect no one else would have published it. Then he didn't hear anything. No one would say to him, "You're crazy," because he's too senior and too respected, but nobody just said anything. And he said that he was giving up on this. By a couple of years later, in 2003, he decided, I guess nobody thinks this is worth pursuing, so, it's like the tree in the forest. If nobody hears you, you don't exist. And then Mike Grecius, who was a resident in neurology at Stanford, and Vinod Menon replicated this default network phenomenon and sent Rakel a paper to edit for PNAS. And he's never looked back. He's totally immersed himself in this phenomenon ever since. And so WashU, which is where Rakel has been his entire career, has really been the driving force of this. But even then, it took a while. 2003 was when that Grecius Menon article was published. But over the last couple of years, this thing has really exploded, and now it's pretty difficult to sort of keep up with every paper that comes out. Most of them are the key didn't come through here, but red is original data in humans. These are reviews, and these are animal models, which is what's starting to sort of take off, because that's really the only way we're going to find out what's the physiologic meaning of this. And here's a recent list, as of the beginning of this year, of the conditions in which this is being applied. And it runs the full gamut, with schizophrenia being at the top and dementia being closely after. And in part, this has to do with the fact that you don't have to get patients to remember to do the task, which is particularly challenging with dementia. But you can record the spontaneous fluctuations of the brain and then relate that to other measures. So let me show you a few examples of what this looks like. This is basically a recapitulation of Biswal's experiment. These squiggles at the bottom are real data from a single subject who's lying in the magnet without moving, and yet one is coming from a blob here and the other coming from a blob there, which are the motor parts of the brain. And they're fluctuating in terms of how much, presumably, neuronal activity is going on there, relatively wildly, and yet very much in synchrony. Right? These two lines are really almost on top of each other. And so this is defined as functional connectivity. In other words, even though these are on opposite sides of the brain, for them to be synchronized means that they have to be talking to each other. maybe directly, maybe indirectly, but they know what the other part is doing. And so this is an example of functional connectivity. And it's measured by measuring the correlation. How much does the variation, when one is going down, does the other one go down? When one is going up, does it go up? So we do that. For example, we can choose this region here, which is where the language circuits are. And if we say that's my area of interest, we'll pull out a snapshot of brain blood flow every 2 seconds, and then we'll pretend that it's connected, and so we'll make a little squiggle that will look sort of like that. This is made-up data, but it doesn't look too far from the real thing. And then, because the computers now can do amazing things, we can say, how about each of the other 100,000 gray matter voxels in the brain? What's going on there? And for each of those, we can get a squiggle. And then we can use very simple statistical methods to pull out where the regions of the brain are correlated, are co-varying together more frequently than you would expect by chance, taking into account that we're looking at 50,000 or 100,000 different voxels, so that we can factor in that a number of those would happen by chance anyways, and there's rules that seem to work pretty well. So at the end of the day, we get a blob, or a series of blobs, that are telling us where in the brain, which regions of the brain are singing in the same, the same song as that one, in a sense. And I like to think of it that it's a little bit like doing nighttime photography, which I love. It just somehow is satisfying to see those colors blend into these rivers of light. And so it's different from fMRI, where you're really trying to capture very specific, discrete events, and different from photography, where you might try and, I was caught by a traffic camera once, and it's surprising how sharp those photographs are. Downright discouraging. This is not a snapshot that gets the driver's license or the registration plate or anything like that. Instead, what you get is the blur of the tail lights or the headlights. And so you can tell where the highway is and where the highway is not. We cannot tell which way things are going. Okay, our correlations could go either way. So we can't tell you if these are tail lights or headlights. But basically, we're detecting where the traffic is, we think. And again, another early paper from the WashU group, in which real data, once more, this time one of those regions of interest that I showed you is placed near the back of the brain. This is called the posterior cingulate cortex, one in the medial prefrontal cortex, yellow versus orange, and those are the tracings. This is about 6.5 minutes. And then this is the midline of the brain. This is the left surface on the side. Intraparietal sulcus is in blue, and its tracings are here in blue. What's intriguing is that the orange and yellow are almost on top of each other. In fact, they're stepping all over each other. The blue is also in counterpoint harmony. I don't think that's the right term, but at least it's clearly synchronized, but in an opposite way. And that is just a fascinating observation, that, again, these brain regions seem to be paying a lot of attention to what the others are doing, even though we used to think that this was so slow that it couldn't be relevant to thinking about what neurons are doing in their millisecond time courses. So around this time, we really sort of immersed ourselves in this, and this is it was actually the first paper we sent out, but it took 9 versions of 3 different journals to finally get it published, and we learned all the things that reviewers were worrying about in the process. But Claire Kelly took a lead on this, and again, it's this Erickson flanker. We were just looking at congruent and incongruence here. 2 different ways of identifying what is described as the default network, this frontal blob and this back blob, with extensions sort of in the lateral and medial temporal lobes. And I'll come back to that a little bit, but that's what Rakel had first talked about in 2001 being a default or a baseline state of the brain, and that network, those regions being the drivers of that. Now, the other words, when we don't have a job to do focusing on something outside, we kind of tune into our internal world and think about how am I feeling, and am I hungry yet, or who knows what. But at any rate, there's this opposite anti-correlated network, which is here in orange, frontal-parietal, related to top-down executive control, to remembering to turn off one's cell phone or things like that. And what was intriguing to us in that these couple of dozen healthy adults is that first of all, the amount of anti-correlation that was there is just striking. This is real data from one subject, one of these people here, and it's almost perfect. It's almost like watching a reflection in a mountain lake before a breeze comes up. You can just see the silhouette of the trees and the mountain peak or whatever. And it's not exactly perfect, but it takes a long time to find where the slight flaws are. And so this is negative 0.98 or so, and the lowest of the correlations was negative 0.82. Now, in part, we asked the computer to find us, what's the most negative correlation. So we were going to get negative correlations because that's what we were asking for. But the range varied, and the range was related to how consistent or inconsistent they were in terms of their response times, which we thought was just terrific. But as I said, it took reviewers a long time to decide that they agreed with that. In the meantime, we were also pursuing this method to find out, what can it do? So we placed regions of interest along the anterior cingulate cortex, 16 of them in each side here, and looked at both the positive relationships, which are mapped in red, and the negative relationships in blue. And so the null hypothesis, the expectation that nothing much was going on here, would have been that this would have been a complete scramble. 6 minutes, no task, this should have been just really uninterpretable. But instead, it turned out to be very systematic. The yellow areas are where the highest correlations are, and those are exactly where the seeds are, where the regions of interest are. But the red and the blue blobs are moving just exactly where the animal studies told us they should. But it's actually quite difficult to see some of these relationships in regular fMRI studies. And one of the reviewers, who turned out was Randy Buckner, asked us, so is this useful in a single subject? And we hadn't tried, but when the reviewer tells you to do something, you always say yes. And so we looked, and in a single subject, we couldn't use whole brain correction there. We got basically less pretty, but the same relationships. And I started jumping up and down after this, because that said that this has the potential to become a clinically relevant index. Anywhere in the brain, we can come up with a number that is true for that person, at least at the time of the scan, that may relate to who they are and how they perform on various other kinds of measures. So one of the first things we had to do was convince Don Klein, who's kind of our advisor and curmudgeon, that this was worth pursuing. And he kept saying, if this is meaningful, if this is real, then it should be stable. And your critics say it's all noise, and noise is not stable, so what's the test-retest reliability? And we kept saying, wait until we get a grant. And he said, you're not going to get a grant until you can show that this is stable. So we got people from Craigslist, we put ads in there saying we were looking for volunteers, and surprisingly, lots of people will answer. We did the best kind of phenotyping we could with no real funding. Then we had them come back, and it turned out to be between 5 and 16 months later. So it was an average of 11 months later, which is not the way you usually set up a test-retest study. Usually you wait a week or so. But this was kind of an extreme just by necessity. To our surprise and delight, it turned out that even a year later, we get reliability values, they're called interclass correlation coefficients, that are as high as 0.9. Now, 1 is perfect, and 1 is almost too perfect. That means it doesn't change at all and it's uninteresting. But we're only showing values greater than 0.5, and so those are what are being mapped in all of these various ways of measuring these measures. As I said, it showed that there's substantial stability over quite substantial periods of time. Since then, other groups have done the more standard 1 week, 2 week, gaps, and again, confirming that these are stable. And I think it's the Stanford group that's also shown that this is stable in children. So again, around that time, we decided to do a pilot study in ADHD, because that's the disorder that I've focused the most on. And we were working with Len Adler, who does medication studies with adults with ADHD. And so we got a number of his referrals before they went into his drug trials. And again, with our Craigslist controls, we had 20 subjects with ADHD and 20 controls. And one of the difficulties, one of the challenges of this technique is that there's 100,000 different ways or more that you can analyze it, and 100,000 different starting points that you could use. And we have to convince people like Cam Carter that we haven't just shopped around and, played fast and loose with our data just to come up with a pretty picture. And the truth is, it happens. Don't tell anyone I told you that, but it's been known to happen, that people will show you, this is what I meant to do all along. I just, I misspoke. So part of what we do is we sort of tie ourselves to the ship's mast in order not to have those temptations. So in this case, we used a paper that had come out a year before by Daniel Weissman and colleagues at Duke. And they reported that there were 3 foci in the brain that were good predictors of whether someone was going to have a long reaction time in about 4 or 5 seconds, those delayed response times. This was in healthy adults, but we thought that's a good place to start. So we used his 3 coordinates, and one of those 3 gave us results, which are shown here. So I think I'm showing you the seed in red, which is in the dorsal anterior cingulate. And when we did that, in the whole sample, we get this positive correlation for that, what's called a task-positive network, and this negative correlation for the front and the back of that default network. But when we compared the 2 groups, they differed where the yellow is being shown. And that is a region that overlies what's called the precuneus and the posterior cingulate cortex. I came into the lab on a Monday morning, and Daniel Margulies had been running these things over the weekend, and he put up a big sign over the lab that said precuneus with an exclamation mark. And all I could tell you about the precuneus that morning was that it was near the cuneus, and that it probably, I vaguely remembered it was occipital, and I think that was it. And but we became big fans of the precuneus. It turned out we weren't the only ones. The very first review that talked about the precuneus had come out a year earlier, in part because the precuneus has among the best blood supply in the brain, which means it's almost never lost in a stroke. And it's very hard to have your precuneus destroyed by a high-velocity bullet that doesn't also kill you. And so there are no patients with isolated precuneus injuries, although it turns out that the posterior cingulate and precuneus is very involved in Alzheimer's disease, which we learned later, in part perhaps because it's got such a high metabolic rate. And so we and others have become very interested in that part of the brain as being a kind of hub to posterior processing centers. So we then use that as a seed or as a starting point for another set of analyses. And again, roughly here, and so you see the same kind of reciprocity that the front and the back of the brains are synchronized, as we would expect, and then these areas are doing the opposite. But here's the ADHD group. Reasonably well consolidated back, but it's like the front of the brain's not talking to the back. And here's where the map of the statistical differences are. Predominantly, well, sort of all over, but especially frontally. So this was also published in Biological Psychiatry. And in fact, we got the acceptance notice about 2 days before submitting a grant that was based on this. which I think made all the difference because that gave it, it's different when it's an accepted paper as opposed to just preliminary data. I was talking with one of your colleagues about Lucina Uden, who was a postdoc in the lab, and so she took that same data set and used a different measure. Here we call it network homogeneity, looking at how correlated are these measures within each network. And it's the same data set, so it should have given us the same results, but that's not necessarily the case. And so we did see nice overlap. Here you see the yellow and the orange are sort of showing where one technique versus the other, and they may not be always exactly on top, but they're pretty close. So again, suggesting that particular region is sort of fairly divergent. So that's sparked us to do a lot of follow-up work, which, as I was telling Cam, we're about to sort of open the gift box, because that's what data always are. You don't know whether you're going to be surprised or not, or if you're going to be able to understand what's in the box. But we're about to look at some data that we've been collecting over the past 3 years or so. But in part, there's been a lot of interest in this default network in a number of disorders. And one of the groups that's done the best work is Randy Buckner's at Harvard. And I love this paper from Jessica Andrews Hanna and his group, in which they combined the best of both techniques, I think, because they did a lot of resting state studies, but they also did some beautiful fMRI. And they had some hypotheses about this, because these regions of the default network have been thought to be involved in self-perception and judgments about oneself. And so they wanted to pin that down. So they asked people who were being scanned to either make judgments about themselves or not themselves, and judgments about themselves in the present or the future. What they found is that these are the two sort of head nodes, that posterior cingulate cortex and the anterior medial prefrontal cortex, here in yellow, and those are the hubs that are connected to all of the other outlying nodes. But the other nodes, the remaining 9, break up nicely into these two sets. These are just called by their anatomic names of some of the seeds, the dorsomedial and the medial temporal subsystems. What was intriguing is that in the task-based fMRI, they found a beautiful dissociation, so that the dorsomedial system was very activated when individuals were making judgments about themselves in the present, but not when they were making judgments about someone else, and not when they were making judgments about themselves in the future. The opposite was true for the medial temporal lobe system. That was strongly activated when people were thinking about, what am I going to do tomorrow, as opposed to thinking about what I'm doing now or how I'm feeling now. So again, I really love that combination. I think that that's where we hope the field will be able to move, is to sort of combine these kinds of methods. But it's also given us a nice set of targets, because they validated these particular coordinates in a number of different data sets. And so again, we use their coordinates so that we're not tempted to move them a little bit or something. And in a recent paper, and I hadn't planned this, Cam, I really hadn't, but another biological psychiatry, maybe subconsciously.
Maybe you're on the edge, right?
I don't think so. But it's been one of the places we like to send some of our good work. We looked at ADHD in 37 kids with ADHD and 37 controls using the Child Behavior Checklist by Tom Achenbach, which is, I used to be very dismissive until I went to Vermont to give grand rounds, and I met with Tom Achenbach, and he was telling me that they have, I can't remember if it's 86 or 89 different languages in which they've made this form available. And when he said that they have 3 forms of relevant to Spanish language, one for Latin American Spanish, one for Castilian Spanish, and one for the Catalans. He had me at the Catalans. Because if you know Spain, you know that Catalunya is not Spain. It's Catalunya. And so this is basically a form that, while it's proprietary, it's not very expensive, and it can be used anywhere in the world. And it's not wedded to the DSM. It was based on factor analyses that have been revised over a number of times and have been based on almost 100,000 subjects by now, so really strong normative database. And so we've been using that as a way of exploring our data. And here, Camille Chaverneau, who was a postdoc with us from France, found a number of different things. So for some relationships and for some measures, it didn't matter whether the children were typically developing controls in these dark dots or ADHD kids in the lighter ones. The same relationship seemed to hold between their score and the strength of the connectivity, which are plotted in these sort of hard-to-see khaki colors here for the posterior intraparietal lobule. And so the relationship between that IPL seed and these medial prefrontal regions is related to, in this case, the total externalizing score, how much of a troublemaker the child is to be non-clinical. Whereas for these, between the hippocampal formation and temporal pole, or the posterior cingulate cortex and the anterior medial prefrontal cortex, the two groups have the same relationships. Again, the greater the score, the greater the connectivity for the internalizing. How badly does the child feel? How sad, depressed, anxious? So for those, we're all dimensional. It's an RDoC world, and diagnosis doesn't matter. And again, same thing true for these. These are all negative relationships. So as connectivity is getting weaker or less or more negative, these are becoming more problematic, more symptomatic. But we also found several relationships where diagnosis did matter, where the ADHD group, or here the typically developing group, between the IPL and the frontal pole, is having a positive relationship between child behavior checklist score and connectivity, and the ADHD group has a negative one, and the same general relationship for temporal pole precuneus. So we concluded that we can't do away with categories yet, that we're going to need to look both dimensionally and categorically, and we think that that's sort of likely to be a general rule that holds up. And the same thing was true when we looked at reading recently. And so here we have healthy readers, unimpaired readers, who are either children or adults. And for a number of relationships, it didn't matter. We chose the seeds from meta-analyses of a number of task-based fMRI studies in children and adults. And so connectivity seems to be related to reading proficiency, regardless of age for some of these. But for others, it mattered whether it was a child or an adult, because children are less masterful than the adults, and so it's much less automatic a process. And so the default network was very much involved in a number of these. So we're continuing to do this work, looking at individual traits. But as I mentioned, one of the things that gets us enthusiastic is that it's relatively easy to collect these data. And so about, 2.5 years ago, we started asking our friends to share their data with us, and kind of setting up a Tom Sawyer thing of whoever shares data by this date gets to be on the paper. We're going to send it to Nature. We did send it to Nature. They declined, and so did Science. But again, PNAS did eventually publish it, and we presented data from 1,093 individuals. And the total cost of the study, besides the postdoc time in my lab, was the dinner that it took to set up the steering committee at the HBM meeting. And this was all sort of data that were already collected, that people were, they already had. And we had no systematizing in terms of how it was collected. We just analyzed it as if it came from the same study, but kept track that it came from different groups. And what came through is that these relationships that I've been describing to you, which can be measured either through the seed-based regression analyses, or you can look at amplitude measures, or you can do independent component analyses, and the list keeps going, were extremely, extremely statistically robust. In fact, the z-scores for these findings are on the order of 50 standard deviation units, extremely significant. And these are the concordances across the 24 centers that were included in these analyses. So, you know, black is perfect, which is what you get when you get the same center with itself, but red is approaching perfection 1.0, and you can see that the means and even the coefficients of variation are highly similar across sites literally around the world. Now, it wasn't true that they were all the same. In fact, the centers differed and in a regionally meaningful way. In other words, it's not that the whole brain is different. These are the maps showing the statistically significant differences between centers, with z-scores going up to 8, which is really pretty reasonable. You know, that's a substantial difference. But when you have a main effect of about 50, you can just factor this out, and we can still detect age and sex effects, which are shown here, so that you can see it's not outliers, but really rather mass sort of population effects that come through even with no prior coordination of how this data were collected. So that's the good news, and we recently wrote a paper called The Promise and Pitfalls of this process. So for example, this shows each of the centers, and the open circles are the 1.5 Tesla scanners, the rest are 3 Tesla scanners, so those weren't clear outliers. But there's a lot of variation, again, because the subjects were different ages, different ethnicities, etc. But it could be modeled because the within-center variance was reasonably good. And so we could just factor that into the regression model. But here's the other piece of the story. Because we had data from 1,093, we could do simulations as if, what if we randomly pulled from those from 10 to 1,000? And the means didn't vary, but the coefficients of, the confidence intervals rather, are ballooning until you get to about 200 per group. And we never get to 200 per group in standard fMRI studies. And so, at least for this technique, it shows us that, you know, we really need to be thinking in terms of good large samples to get definitive results. And so again, that's part of the motivation for encouraging people to share more data. And so we love new acronyms and logos. So this is one of our newest ones, the International Neuroimaging Data Sharing Initiative. And we require that people get ethical approval locally. We take the face off and make sure that they're anonymized, so that, because these data are made available publicly in open repositories. And but we've made these data available, and they've been downloaded thousands of times, more than 20,000 times, I think. And one of the first to do so was Nora Volkow. And she was sick for a week, and so she downloaded our data to her laptop, and she and Dardo wrote a paper that week. So they were the first out of the gate. And she was blown away. Again, this is using Dardo's measure of functional connectivity density mapping, that with no coordination at all, you can basically see the same pattern across the board in every site. And she went from being a skeptic to being a strong proponent of this. So more recently, we started at the Nathan Klein, where I also work, to collect data which grew from an initial small pilot to eventually 250 people who were assessed. We had imaging data from 207, which we gave away every week as we were collecting it. And that prompted the NIH to fund Mike Millam to do this more and better. And so he's in the process of doing that. So this is showing what's available now from the already collected 207 subjects, but there's an even larger, more extensive 2-day phenotyping that's being done with the next set, which will be representative basically of the US population, and go from age 5 or so to 80. More recently, we created this ADHD 200. It was supposed to be 100 ADHD kids and 100 controls. We sort of overshot. We put 285 ADHD data sets and almost 500 controls up on the website in March last year. And of course, Nora was the first to publish from that data set. And again, showing interesting differences in local connectivity relating to orbital frontal and reward ventral striatal regions, and frontal parietal decreased connectivity in long-distance relationships. So we really are encouraging investigators to, at the very least, contribute their data once they've published 1, 2, or 3 papers, because that's about as many as we do. And we all have essentially suitcases worth of digital data that just get old and die. We never get back to our data that we think we're going to, and it's much better to make it available to the community. There is the hidden shame of what if someone finds a mistake, but we think that those are inevitable, and the sooner the better, and that we need to sort of get used to that idea. So we also think that people should be providing their scripts so that we don't have to guess and try and figure out why the results are different from ours, and that if we have the same data to work with, the same scripts, we all should be getting the same result or something's wrong, and then we can work out those things and really make this work as a community. Because the goal is to use this to basically get to high technology, which is what this is. These are the growth charts. Every person who has a child has seen their child plotted on a growth chart. And it doesn't look like much, but just by measuring the height, weight, head circumference, and plotting it on one of these graphs, the nurse or the pediatrician is doing millions of dollars worth of workup, or avoiding millions of dollars worth of workup, because you can rule out 2,000 different causes of poor growth by simply knowing that the growth is proceeding as expected. And if it's not, then you have to work out and find out why. So this is what Lewis Thomas would have called high technology, and we have the wherewithal to get to that with the brain. The first example of that was published again by WashU a year and a half ago. It's called Functional Connectivity Multivariate Pattern Analysis, which uses machine learning and computer methods to create hyperspace differentiations, etc. But basically, you can take these connectivities and boil them down to an index that relates to the maturation of the brain. And that's what they did here as a proof of concept. We've done similar work with much smaller samples, with about a dozen or so children, adolescents, and adults, and again showed what people find, which is that children have greater density of correlations nearby, but they don't have the long distance relationships, and those start to emerge in adolescence and become more prominent in adulthood. So the brain is becoming more integrated as we mature, and so it's a bunch of local neighborhoods in a child. And if you've ever talked to a child who's an expert on Harry Potter, they'll know more about Harry Potter than you'll ever know, but they're not going to talk about the allegorical meaning. That's not what they want to hear about. So let me conclude by, I've just touched on this notion, but I really do think that inconsistency is both a clinical clue and a neuropsychological feature that we can get a handle on. And it's related to genetic susceptibilities, and imaging can use that also to learn more about what's going in the brain. The default network hypothesis really is that one of the problems in ADHD is likely to be a faulty interregulation between the default network and these other control networks, and that works most of the time, but sometimes it really fails intermittently, and when that happens, it's kind of a brownout situation, and so there's a lapse of attention, and if that happens in math class, you could be in real trouble, because all of a sudden you missed a key concept, and you don't know how to create the intellectual structure that the teacher's hoping that you'll be building inside your own brain. And I haven't shown you much, and the data are really still fairly scant, but there have been 3 or 4 studies that have been coming from other groups that have been supporting that, especially in terms of medication effects that seem to be consolidating or solidifying some of those relationships. So time will tell whether or not that really gives us a beginning of a physiologic understanding of one of the mechanisms that underlies some of the difficulties in attention and in behavioral regulation. It's unlikely to be only relevant to ADHD. It's very likely to cross a number of different boundaries, and again, it gets us back to the RDoC idea. So, as you can tell, I'm pretty enthusiastic about the ability to image the brain without a task, so-called resting state, but there are a lot of pitfalls as well, but by harnessing the brain power that we have and the data that we've collected, we think we can have a model where we accelerate the rate at which we work through some of these problems. What's beautiful about this is that it works in animals, it works in humans, it works with an anesthesia, everything has to be taken into account, but I'm working on a rat study right now, and we're seeing effects of aging and development in the rat and of the poverty of the mother, so that if a rat is given less bedding to make a nest, it becomes very stressed, and so it's much harsher with its pups, and then those pups, when they grow up, become more like depressed adults. And we can see the differences in amygdala connectivity, even though the rat's anesthetized while we're scanning. So we'll be sending that out for publication, I hope, in the next month or so. A lot of challenges to be dealt with, but many of these can really be addressed by being more open about our data, and the NIH is supporting that in a big way. The Human Connectome Project received $40 million to provide data on a twin sample with genetics and the super high-tech state-of-the-art imaging, and those will be public data that everyone gets a chance to kind of understand and embed, because again, if every person in this room started working on it, we wouldn't write all the papers that are available in any single one data set. There's a lot of juice that's left in these data sets that we don't squeeze out. So with that, let me finish and say that Mike Millam has been really the CEO of the lab for the last 5 years. He's now doing that, as well as set up his own independent lab at the Child Mind Institute, which is across town, and we also work together at the Nathan Klein. Claire Kelly is now sort of the one who's leading the imaging in our group, and Adriana DiMartino is also leading all the clinical efforts and the imaging in autism and ADHD, along with even longer list of individuals who have passed through. So thank you very much for your invitation, your attention, and I'll be happy to take questions.
So it stands to reason from a lot your discussion was that this variability is really a predictor of inattention. But there's been a number of studies that also shows that it correlates with hyperactivity and impulsivity. So I was wondering if you could speculate upon the relationship between inter-individual variability and those other features of ADHD.
So that's, again, ADHD I find fascinating. And there's good evidence that inattention on the one hand and hyperactivity and impulsivity on the other hand can be separated as dimensions. But even when they're separated, they're very highly correlated, of about correlation coefficient of about 0.7. And there also seems to be a more general dimension that is related to ADHD-ness. And so part of what we'll have to work out, and I think it's going to take large samples to do this, is what goes with what in what part of the brain. So it's conceivable that the entire brain is doing the same thing, but it's more likely that there'll be certain regions that are more related to certain kinds of behaviors. So the supplementary motor area, for example, the motor system may well be related to some of the hyperactivity, and maybe more frontal parietal regions, ventral and dorsal attentional areas, precuneus are going to be related to inattention. I mean, those are just possibilities, but I think we can test those, and if we do it with this sort of approach, we'll always have to remember that initial findings are hypotheses to be verified. And so by accumulating large data sets, we can break them up into two sets and then use one for discovery of relationships and the other for replication, and use that as a filter to really only focus on those that survive that. Because the statistical techniques are such that we'll always find some things, and the challenge is figuring out which ones are worth further exploration or pursuit. So we found that kind of replication does a lot of winnowing down and gets us to focus our attention on the more, the higher probability relationships. So, as I said, I'm hoping that we'll have some initial answers to these questions by the end of the summer, because we've been collecting data for the last several years, and it's certainly something we're going to want to look at.
Thank you. The idea, I believe, to uphold which the medical industry would uphold, we do no harm. And to thinking about that, why don't you advocate giving ADHD kids omega-3 and omega-6 along with changing their diet, along with instead of these toxic pharmaceuticals and stimulants? I just noticed you cite experiments which were really just utter torture of humans and animals. And but you also treat it like the brain is like a computer rather than you can diagnose it like a computer rather than it's a unique creation of God.
Well, the brain is not like a computer. A computer is fairly inflexible, and one of the wonders about the brain is that it's extremely flexible and mysterious. And so we are forced to take things in a very simple-minded way, just because this is arguably, this makes rocket science look easy. But I think omega-3 is probably a good thing. But it's not easy to find evidence that tells us how much and what type people should be taking. The effects are modest. I personally take omega-3 because I think why not? I have no problem with that. But there's a lot of need for better across the board. And I appreciate your intensity and your dedication. But I suspect that the approaches that I take would be objectionable to, on fundamental levels, and we'll probably just have to agree to disagree about those.
I was wondering whether there have been any studies comparing between disorders in terms of, because I know there's been some people in your group you mentioned also are looking at autism. Mm-hmm. So I was wondering if there's been actually comparisons and if people who have overlapping symptoms of the ADHD as well as some of the symptoms of autism.
So, as Julie said, we've been working on a study where we recruit children who have ADHD, obviously, or we've expanded that sample because we've already been doing that. And then we've recruited children with autism, about 60% of whom would otherwise meet criteria for ADHD, and about 40% who don't. And obviously typically developing children for comparison. So those analyses are also going on as I speak, probably, literally today, this week, next week. But Adriana DiMartino has been taking a lead on that. And the initial look when she was preparing an abstract a few weeks ago were that, at least with ADHD, putamen centrality or centrality measures in the putamen were deviant in children with ADHD or children with ASD and ADHD symptoms. It didn't matter whether or not they had ASD, and different from the typically developing children, obviously. So that was an intriguing finding. There were some other findings relating to amygdala centrality that were a bit more on the edge, and so she's expanded the sample, and we'll see whether or not they reemerge because they were coming and going. That seemed to be related to ASD and not ADHD. It'd be nice to have a dissociation in both of those. But clinically, we're finding that in terms of the usual rating scales and measures, the ADHD in the ASD children looks very much like the ADHD in the ADHD children. And we thought, it might be more atypical. Part of this may relate to our need for most of our studies to study children who are high functioning. So we have an IQ minimum of 80, because otherwise it becomes difficult for them to understand what's going on and for them to stay sufficiently still on the magnet to give us measurable data. And that may be affecting the kind of ADHD that we see. But it looks more like garden variety ADHD, very commonly, obviously, in those kids. And as an aside, the DSM work group on ADHD is taken a position, which we've been explicit about, that we're going to remove the exclusion of autism spectrum disorders, so that if one meets criteria for ADHD, they will get that diagnosis as well. And that's been at the very express request of the autism community, because it's been a real frustration for many of them. But it was interesting, and not exactly what I expected. I thought we'd see some deviations, but we've just started to look. So we'll look for connections in which they diverge as well.
My question is on phenotype, inattentive type of ADHD. You commented on that, the interrelationship between those two phenotypes. But can you just focus on inattentive type for a moment, and how this particular ADHD phenotype evolves differently as we look at the DSM progression from 3, 4, 5?
Well, that's one of the difficulties, is that DSM, keeps changing, and so everything sort of becomes obsolete, and you have to kind of start over. And so I'm not sure if you're asking about the DSM part of this or what's going on with the DSM. I'm going to give you an opinion. I have 1 vote out of 8, so I can't promise that things will go my way. But I suspect the only change we'll make with regard to inattentive is that instead of calling it a subtype, we'll call it a presentation. It's a small semantic difference, but there's enough data that the so-called subtypes are, they don't stay the same. And so there may be some context differences. It may be that it's measurement error, but it's fairly compelling that you can't make an assumption about someone if they're inattentive once, they're inattentive forever. In fact, a number of individuals who are combined type sort of morph into inattentive types as they become more mature, or their environments are different and they don't have to sit in a classroom all day, et cetera. So my guess, as I said, is that will really be the only functional or the only relevant change. I think we are changing the age of onset requirement to 12, and we've written a commentary about that a couple of years ago, based on our best guess of having to have an arbitrary cut point at some age, but deciding that 7 was too young. And we're adding examples of the criteria, and I think that will probably go into the new version. But I suspect we'll keep the 18 criteria otherwise pretty much as they are, because they work, and we don't know how to change the cut points or the requirements if we add more criteria, and if we don't know what's going to happen if we change some of the criteria for the newer ones. There's just too much uncertainty, so we're going to need to wait for future iterations. And one of the things that's been pointed out repeatedly is that this DSM will abandon the Roman numeral tradition. It's going to be an Arabic 5. And I keep saying that we should just go ahead and call it 5.0, but they won't do that because that sounds too downbeat. But there is a promise, or at least the exhortation or the hope, that we'll have decimal iterations, so that we won't be waiting 15, 20 years for DSM-6, but rather we'll have DSM 5.1 and 5.2 and 5.3 and so on and so forth as we learn more and we understand better what distinctions really matter. And I think that there's a real likelihood that that's going to emerge more and more. Progress in science is nonlinear. It takes a really long time to seem to not be making any progress, and then all of a sudden things explode. And so I'm kind of an optimist about that, and I think that there's a real chance to understand pathophysiology. Genetics isn't going to give us diagnosis. That was always kind of a silly idea. But it gives us some of the mechanisms and some of the molecules that are involved. And then if we can identify where those molecules are having their greatest impact through imaging, we can start to make models of what the physiology is that's underlying some of these disorders. And it's through that understanding that we get to real diagnostic sophistication. As a pediatrician, I don't practice anymore as a pediatrician, or I only did it as a resident, but telling me that a child has a fever means nothing. If it's, a 1-month-old, that's a life or death emergency, and I'm going to be injecting antibiotics before I do anything. If it's a 7-year-old, take 2 Tylenol and let's see what happens. If it's a child with a viral syndrome, if it's a child with rheumatoid arthritis or leukemia, etc., all of those matter because fever is just a number that tells us that something isn't going on right, but there's a number of different causal situations that could be going on, and a pediatrician has to make hypotheses about those and has a whole way of working through what could be going on. And so I hope that we will eventually get to that place where we can start to use these high-tech tools to understand, not every child should get a scan, but there's a lot of kids about whom we really are uncertain. What's primary, what's a compensation, what's, is this truly autism with decreased intellectual ability, or is it basically intellectual deficiency with difficulties in a number of areas, for example? That's a big question from what I understand. And I think that we should be able to get brain signatures that will help us to understand that, as many of you are working on some of the treatable molecules that can be affected by sometimes antibiotics or statins have effects on brain function, for crying out loud, in certain ways. I mean, we start to have the potential to really have impact, and as was just said, we have to think about do no harm. If we're going to change the brain, we could make it worse, and so we have to be judicious. But being nihilistic and saying it's genetic and so it's done, doesn't work anymore. We know that the brain is sort of continuously reinventing itself and adapting, and so if we can find the fulcrum points, the places where a small amount of influence could have a larger effect, there's the real potential to intervene in ways that are meaningful. Again, the more we do this, looking over each other's shoulder and learning from each other, the more rapidly we get to that.
I was wondering if you could talk a little bit about how your default network hypothesis relates to some other common ideas like executive dysfunction and dysfunction in reward systems.
Let me, free associate a bit. I think that, again, there's a number of factors that are likely involved. And so there's a surprising amount of convergence. It's only 3 or 4 studies, but when you see 3 or 4 studies that are lining up, it's not nothing. That reward circuits are aberrant in ADHD, and one of the questions is whether that's true across the board, or is that more related to conduct disorder types of ADHD? We don't know. We haven't looked at that systematically. Again, one of the difficulties is that studies in general have been small, and so samples of I've published a study with 11 kids in a group, which doesn't get published anymore, but even 20 is really fairly small. And so to look at these issues of heterogeneity, we need to have larger samples, and these are expensive things. And so it's another reason why I think we can do some of this with larger aggregated data sets. Executive function measures, The effect sizes are not trivial, but they're not huge. And one of the questions I have is, is that a primary problem, or is it an issue of the inconsistency? And to a certain extent, it's not true in every case, but in tasks like stop task, which maybe is not an optimal executive function task, when you co-vary for the variability, in many cases the group effects kind of wash out. And so it suggests that the inconsistency of responding is being interpreted as an inhibitory deficiency, perhaps. So I think that attending to those and trying to work out, is this, is it impervious to that, or is it really an inability to do this performance, or an inability to sustain it, which I think is more likely. That I think does fit in with this default network kind of intermittent hypothesis that why is the default network so insistent on maintaining its fluctuations and having this kind of partnership with executive control networks? I really think that that's an important clue. There's a study that Jim Chi did in healthy young adults, either after a full night's sleep or after 24 hours without sleeping. And without sleeping, they looked like ADHD individuals. They made lots of mistakes on cognitive tasks, and the variability was much greater. People are impulsive, etc. And their default network, the front and the posterior nodes were less well correlated, and also the anti-correlation with the frontal parietal networks was decreased in magnitude. And so I think both of those speak to the brain kind of liking this yin-yang intermittent sort of approach, and that being maybe an optimal state for functioning. We know that when individuals are doing cognitive tasks, the amount of default network excursion decreases, but it never decreases to zero. It still does some fluctuation, but it's quieter. And then, when they kind of escape, in a sense, and start thinking about something else, frequently that's associated with default network non-suppression. So it seems to have some interesting reality, but it's there in rats, it's there in monkeys, it's a part of being a mammalian brain, apparently. And so understanding what it's doing and why it's so insistent is one of the things that we're curious about. But I think that taking into account, taking the temporal, taking time seriously will help us to understand executive function and cognition better. In part because of the need to average, whether it's an fMRI or an ERP or in cognitive tasks, we've collapsed the dimension of time. We lose other things with resting state, but we maintain the temporal structure. And so that's sort of what's up, I think, is to reconceive of some of these phenomena, taking this background into account.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.
And in this case, there's really no particular frequency band that stands out in this child's record, whereas in the ADHD child, there's a very clear peak at about a 20th of a hertz. In other words, it takes about 20 seconds for one of these cycles to go through its full up and down oscillation. But when that child was given methylphenidate, you see the darker tracings, and you see the tracing of the frequency analysis. In other words, that variability was abolished and brought to the control levels by Ritalin brand or methylphenidate. And so that struck us as interesting. And around that same time, there was a group in Germany, Christoph Klein and colleagues published several measures, go, no go, stop task, continuous performance task, and NBAC. And regardless of the task, the variability of the children's response times, measured in a couple different ways, was always the best way of discriminating the children with ADHD from the controls. And in fact, when statistical analyses were done, called principal component analyses, about 2/3 of the variation in response times could be assigned to this single focus of variability. Jona Kunze and her colleagues at the Institute of Psychiatry have done a series of studies going back to 2001 showing that, in fact, this is the only neuropsychological trait that seems to be heritable and very robustly heritable. So again, it speaks to genes being involved in how it manifests. And what's interesting is that this kind of variation in response times frequently masks as the inability to do executive function tasks, because what you get is less average performance, and it looks like they don't get it, but they get it very well, and then they don't get it periodically.
This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs. Also, make sure to check out and subscribe to our YouTube original channel, UCTV Prime, available only on YouTube.
I'll talk a little bit about one aspect of ADHD that has seemed to us to be an entryway into the confusion that is this common disorder. And we'll focus mostly on the use of what's commonly called resting state, but that's probably a misnomer, but a mode of measuring brain function without a task that seems to be giving us some exciting clues. And one of those has to do with what's called the default network interference hypothesis. And one of the ways in which we're exploring this is to aggregate samples from investigators literally around the world. And I'd like to propose that that's something that we should look forward to doing more and more. So about 10 years ago, Rosemary Tannock and I were musing about what did we understand about ADHD. And we're thinking about the decades of neuropsychological work that have been done trying to get to a test. Everyone wants some kind of objective marker, some way of measuring that would be either like a blood test or a blood pressure test or something that doesn't rely simply on opinions. And it's been 30, 40 years of disappointing work. And in part, that has to do with a standard Erickson flanker task in kids with ADHD, where they're told just to pay attention to this arrow in the middle and ignore the others. But sometimes the arrows are all pointing in the same direction, other times they're conflicting, and other times there's just a rectangle that doesn't give you any information. And what's nice about this test is that you can predict very well that individuals, it's easy to do, first of all, you just have to say left or right over here, but it takes between 50 to 100 milliseconds more on average to respond to these stimuli than it does to that one. So you can really tell if people are sort of tracking the task or not. And Anouk was getting the kids to do this every 3 seconds. And so this seemed like a good place to examine consistency and see whether or not that was systematically varying. And here's a couple of illustrative cases. This is what a control looks like, and we've done a little bit of smoothing here, but more or less there's going to be some up and down jitter because, again, it takes longer to respond to those conflicting trials than to the non-conflicting ones. But here's a typical child with ADHD. On average, they're about the same, about half a second. But here, this one took almost 3 times longer to do that same task. And here it's twice as long, and there it's almost twice as long. So those we take as indicators of lapses of attention, that the child knows every 3 seconds I'm going to have to respond, but occasionally they're having to bring themselves back sort of from a longer distance. Now, these data can be analyzed in a number of different ways, and one of those is by assuming that, in a sense, it's like a piece of music. And so we can do a frequency analysis, which is the same thing that happens when people package music into an MP3 file or a CD, which is you break up the musical spectrum into frequency bands. And these are the frequency bands along the bottom axis. And then you can determine how intense or how loud the frequencies are at any given frequency with the dramatically substantial inconsistency of performance of individuals with ADHD who will do beautifully on a test one day and terribly the next, or even that same day for part of the time. They'll go in and out. And so we thought about how that inconsistency perhaps was an important clue. And we suggested that not based only on our own observations, obviously, but on previous literature that had started to highlight that. And one of the things about that concept is that it's relatively easily measured if we track how long it takes someone to push a button, response time. And there's a finite amount of time. You can't respond in 0 time because it takes about a quarter of a second for the information on the computer screen to get through your eyes to the visual part of your brain, and then for that information to be processed and eventually make it through to the motor centers, etc. So there's always a minimum amount of time required, but there isn't always a maximum amount of time required. It could take longer. If you're distracted by a cell phone ringing, then you come back to what you were doing, then it'll take longer on that trial than in other trials. And so when we measure a series of response times and sort of put them up next to each other, we typically see a kind of ski hill configuration. So it's sort of steep on this end and then long and sloping on that end. And what a number of investigators had been observing is that the ski hill was longer for individuals with ADHD. So they would tend to have more of these very long, prolonged response times. And this was true from data aggregated from the MTA study, which was a large multi-site study. And we also found this in data from a postdoc at the time, and Auxiers, who had used periodically. And so we've been chasing that idea down. And in biological psychiatry, we published a paper in 2008 in which we took an independent sample and gave them the same task, and again measured those frequency differences and found that there were more variation, especially in that very low frequency range in the children with ADHD, and that the more of that energy they had, the more likely they were to make various kinds of errors, here omission errors. Catherine Johnson at Trinity College in Dublin had found a similar observation in about half the kids with ADHD. The ones that were impaired in terms of making more errors had clearly greater intensity of that low frequency variation, and with different kinds of task. It's called a sustained attention response task. And so we recently did both that SART and the Erickson flanker. This is in a group of Italian children, and again found that using both methods of measuring frequencies. I won't go into the technical issues, but one's Morley wavelet-based, and the other is the Fourier frequency transform. In both cases, for both tasks, we see greater variability in the individuals with ADHD than the controls. Many of these kids unmedicated ever. And again, when we separate them out by the impaired versus the unimpaired, we see that some kids with ADHD look just like the controls, and others are very substantially impaired, these green sort of shadings there. We're not the only ones. Again, the Johnson's group and others have found this. Not everyone gets it, so there are issues perhaps about tasks, etc. But it's compelling enough that we've been able to proceed with this. And around the same time, in the world of neuroscience, Yuri Busaki and his colleagues, who were really interested in oscillations